so that everybody understands it. Like 10 years ago, approximately, I moved to Munich, and I started working in this subject as a postdoc, and now we moved to Zurich, and we continue working on the same. I think it's a very nice topic, and I'd like to give you an overview of what we're doing. So it's about optoacoustic imaging. It's a new imaging modality that it's very advantageous, and I'll try to convince you that it's very nice to do research on it. So probably most of you didn't hear or haven't heard about this technique in your life. So one thing that we can do is to search for it, for example, in Web of Science. It's called optoacoustics sometimes it's called photoacoustics. Some groups call it one way, some groups other ways, just political reasons, but it's exactly the same. So if we look at it and we have a look at, for example, of the number of papers and number of citations, so you see that the trend is going up. It went a bit down last year, but it's probably because of the COVID. And probably the first paper that really pushed the technique forward was this paper in 2003, which was published in Nature Biotechnology, where it was shown for the first time that optoacoustics can do something useful, in this case, detect brain activation in rats. And since that moment, you see that the trend is exponentially growing, and there is a reason for that. So, so it's very difficult to find this trend in any imaging modality. So you can imagine that it must have something good. Okay, so I'll talk about technical aspects, but first I'd like to give an overview. Probably many of you have never worked with imaging modalities. So there are actually many approaches, many technologies to see inside the human body or inside the body of an animal. And each provides a different advantage. So, and you cannot just have a look at how the images look like, because, for example, here, this is an example of two images with PET and with MRI. And probably if I ask you which image you prefer, most of you would choose the image on the right. But if you have a look at what the images provide, and if you superimpose it here, this is a graph that I generally take. It's from a professor from Stanford that sadly passed away recently. But he generally shows this to show the importance of early stage of early detection of cancer. So if you detect the cancer at the first stage, it's most likely that you the patient survives, and if you detect it at the later stage, probably it's too late. And even though the image in the right, you see that it's very nice, if you want to detect a tumor based on the anatomy, which is what you see in the image of the right, this is generally a late stage. However, the image in the left shows something different. It shows functional and molecular information, shows some disease cells that have different undergo different processes, different molecular pathways, and these are detected in this case with FDG, which is a tracer, a PET tracer. So you can not only look at the anatomy, which is what you generally think of when you think about imaging, but there are other things that imaging modality can provide. There's also genetic information. This is an image of green fluorescence protein. There was a Nobel Prize about this. And with this information, you can also, you can label cells very specifically. You can do many things. You can study biology at a fundamental level. And here you can see also one of the limitations of optical imaging. That is, if you image something superficial, it's very nice, but if you try to go deep into tissue, everything is diffuse. The information is there, but it's just that you cannot resolve it. And of course, there is also dynamics. So biology is very dynamic, and it's not only providing an image, but see how that image changes with time to see how, in this case, it's a beating heart visualized with ultrasound, but there are many other biological processes that are dynamic. Basically, anything in vivo is dynamic. So I'll try to convince you that optoacoustics can provide most of these things, or almost everything. Of course, there are some drawbacks, but it has it provides a lot of information. Why does it provide such an amount of information? Well, we like to say that optoacoustics is a 5-dimensional optoacoustic imaging modality. So we have 5 different dimensions to provide information. And this is illustrated, for example, in this image here. This is actually a video that I'm going to play. I hope it works. So this is an image of my hands. So here in the upper part of the video, you see that this is basically how the optoacoustic image looks like. What you mainly see is blood vessels, it's what mainly absorbs light into tissues. And you see that, we can provide the 3D image, but we can also change the wavelength, meaning that we have an additional information, the wavelength content. And then if we do some processing, we can unmix, for example, oxygenated hemoglobin from deoxygenated, and also melanin that you see here, skin pigmentation in brown in my skin and in yellow in the images. So we have, on the one hand, we have a 3D imaging modality. We have also a time dimension, because this is real time, and we have also this spectral dimension. So that's what we call 5-dimensional optoacoustic imaging. And this is a lot of information that we can process, and I'll try to guide you a bit on how to do it, and from the very basic to the most recent things that we're doing. Okay, so basically, let's start from the very basic. So what do we need to build an optoacoustic image, or how does it work at a fundamental level? So optoacoustics, as the name indicates, is a mixture between optics and ultrasound. The optical part is related to the excitation of tissue. So we use a laser to excite tissue. This laser will induce thermal expansion in the tissues, will generate a local pressure increase, and this will relax in the form of an acoustic wave. So we emit a wave, or we generate a wave inside the tissue, and this wave is detected outside with an ultrasound transducer. So the acoustic part is this ultrasound part, is this ultrasound propagation, basically. So we have light in and ultrasound out. So this is what opto is what goes in, acoustic is what goes out. So this is a small animation. We have a laser heats up the tissue and then generates an ultrasound wave, and we have a signal like that. So this signal is very similar to what we get in ultrasound. For example, is a time resolved signal, it's a pressure signal. In ultrasound, they call it the RF signal or a mode signal, or there are different names, but it's just basically pressure as a function of time. It's an ultrasound wave that you detect. So what do we need to build the system? So we need a light source. Generally, it's a short pulse laser, but there are also other types of light sources. We need to detect the ultrasound, so we need an ultrasound sensor plus the data acquisition system. And we need, of course, also some mathematical modeling, because we need to reconstruct an image and build, to provide imaging from the raw data, we need to process it, of course. So we need hardware component, and also software component is essential. Okay, so the other thing that is, I think, important to take into account is that it's as a relatively new modality, but not only because of that, there are actually many different implementations of optoacoustics. You see here, this is an example from a review in 2016 in Nature Methods, where you see that, there are 8 completely different systems here, but there are actually many more. And so optoacoustics is not really like ultrasound that you think how the scanner looks like an MRI machine, whatever. You have a clear picture of how a system looks like. In optoacoustics, it's not like this. All these systems are completely different, and all of them are needed because they're used for different applications and in different regimes, different penetration depths, different ranges in general. Okay, so, and then of course it's the software part that we have. On the one hand, the signals, which, as I mentioned, they're ultrasound signals, and then we have to do something which is generally reconstruction algorithm plus something else to get final image. So this something generally just to split it into 3 different parts. So we have, on the one hand, we need to do some signal processing, of course, and we need to apply a reconstruction algorithm. And finally, when we have the images, particularly when we have the multiple images at multiple wavelengths, we can also process them with the additional algorithms to extract information out of them. Okay, so I'll try to, as I mentioned, explain you a bit the different implementations of the technology. So the simplest thing that we can do is what's called optical resolution microscopy, or optical resolution optoacoustic microscopy. And the very basic idea is to take advantage of the fact that, you can focus light, which is like the way imaging is done, for example, in scanning optical microscopy. So basically, one possibility is to focus light and then move the focus spot in raster scan configuration. And basically, the signal at every point would be your pixel in the image. The problem is that this we can only do in the first millimeter in tissues, because at higher depths, you see here what happens. Light starts scattering inside the tissues, and then it gets diffused. So that's the reason why we cannot see deep with optics. Everything appears blurred. It's like trying to image through the fog. But if we stay in this first millimeter, we can get very nice images simply by focusing light and scanning it around. So this is very simple implementation. It's like scanning optical microscopy. with the additional advantage, we have also some depth information. Also, depth is not that important in this implementation, but we can basically just format 2D image by putting together the acquired signals at different scanning positions. And you get an image like this, which, as I mentioned, is an image of the blood vessels, an image of the vasculature, because what mainly absorbs light in tissues is hemoglobin in blood. And this is why we see the blood vessels mainly in optoacoustic images. Okay, so this, as I mentioned, is the easiest thing to implement, but we're not really doing much about this, at least not in the projects in which I'm involved. So what we're mainly interested is to go deeper than this, because is where optoacoustics can become more powerful, because you can see something superficial only with fluorescence or generally with optical microscopy, but if you want to go deeper, light gets diffused and you cannot image anything, or not with sufficient resolution. But if you take advantage of the fact that the ultrasound doesn't scatter so much as light, then you can do an image. So one possibility is to use a focus transducer in a configuration that is generally called acoustic-resolution optoacoustic microscopy. And then basically it doesn't matter that light gets diffused or gets scattered everywhere, because like the sensitivity is confined by ultrasound. So you can, I don't know if you can see my mouse, but everything okay? Yeah, okay. So yeah, basically, if you use a transducer like this, you would detect signals mainly from this line here and not from anywhere else. So you have this selectivity given by ultrasound or by ultrasound focusing, regardless of what light does. Light can get very diffuse, but we have the resolution given by ultrasound. And then again, we can raster scan and get an image like this, which in the first approximation, we can, of course, just put together the signals, but we can do some modeling. And we can take into account of, well, the very basic principle of optoacoustic generation, which is well described mathematically. And then in this case, it's also important to take into account the response of the transducer, which is what gives us the selectivity. And this is generally divided into two terms. One is what's called the electrical impulse response, and the other is the spatial impulse response. So basically, the signal that we detect with the transducer is like the pressure signal combined with these two terms. So if we use this information, we can also develop a reconstruction algorithm that is better than just putting together the signals. And the other configuration of optoacoustics is what's called, well, as I mentioned, there are actually many configurations, and we can talk about here for long, but the main configurations, let's say, the other main configurations is what's called optoacoustic tomography. And here the idea is not to use a focus light or a focus transducer, but rather to acquire optoacoustic signals from different positions around the imaging, the sample to be imaged, basically. And then in this case, it's essential. I mean, there's no way to get an image without any reconstruction algorithm. So in the other cases, I mentioned that, okay, we can, as a first approximation, just put together the signals at different scanning positions. But here we need some mathematics, we need some processing to get an image. So yeah. So here in the case of to design a proper optoacoustic tomography system, we have to take into account one thing is the acquisition geometry. I briefly comment on it in the next slide, and the other, of course, is the reconstruction algorithm. So both things are very important to get a good optoacoustic image. Okay, so why is it important, the acquisition geometry? So this is an example that from a paper that we published back in 2016. So if we think of ultrasound, generally, what we have is an ultrasound array, which is generally, here is like an arc, but generally it's a linear array, right? And it's only in one side of the object. And if we think of an ultrasound image, what we generally get is an image like this one that you see in the image, which is what's called a speckle image. Or we have these speckle artifacts everywhere, these kind of dotted structures when we do ultrasound. The thing is, we don't have this in optoacoustics, or we don't have it when we have like a dense distribution of sources, like we have, for example, in blood. The individual sources would be red blood cells. Okay, but this has a good part and a negative part, because if you compare these two images, for example, you would see that when we have a speckle image, we have an image that we have this noise, these dotted structures, but overall we see most of the structures in the image. But in optoacoustics, we see that okay, for some orientations of this thing that it mimics a blood vessel in this case, we see it very nicely, but we're missing half of the image. And the reason of that is that we don't have this speckle, but we have a problem that is called limited view visibility. Optoacoustics is inherently a tomographic modality, and then if we want to reconstruct it properly, we need to acquire information from all around the object. We need to get like at least 180 degrees. In this case, this is an image that we acquired with 270. I mean, this is obviously just a simulation to show the very basic principle. But if we get more than 180 degrees, you see that we recover properly the image. But if we have less than 180 degrees, the image is strongly distorted. So this is essentially if we want to make a good optoacoustic image. The problem is that this is not always possible, especially if we want to use, for example, optoacoustics in the clinics where we can only access the body from one side, not from two sides. Yeah, so this is a fundamental problem in optoacoustic images that is still ongoing research on it. Okay, so as I mentioned, optoacoustics is inherently tomographically. We need this information all around the object to get a good image. And this is a well-known problem, for example, in X-ray CT, where we have the mathematical model. And then from that mathematical model, we can derive the inversion formula, which is what's called the back projection formula. So there is something analogous in optoacoustics, basically, which is also called the back projection or filter back projection algorithm in optoacoustics. And basically, it's the same thing. So when we have, so this on the top would be an X-ray image, and on the bottom we would see an optoacoustic image with one of our systems. Yeah. But this is, let's say, the analytical solution of this equation, which is not always possible to apply, but generally it works pretty nicely. So this is, I would say, the standard method of doing reconstruction in optoacoustics, which is the same as X-ray CT. It's not exactly the same, but the same idea, let's say. But of course, this is not only the only way to do reconstruction. We can also do what we call model-based reconstruction, which is based on basically taking the like the linear model, which is a continuous linear model that describes optoacoustic generation, and discretize it to make like a matrix equation that you see here. So you have like the theoretical pressures given by the product of a model matrix times the P0 would be like your image. What you're reconstructing in an optoacoustic image is the initial pressure, which is proportional to the absorption. And then we can numerically invert this problem and get an image. So this was done. I was not even in, well, I just arrived recently at the institute back then. But this is, I think, a nice example to compare the result that we obtain with back projection and with a model based in a two-dimensional system, which is this system that you see here. Two-dimensional system is basically built by having a transducer, an array of transducers that are focused in one plane. So we only get an image of basically, in this case, of a section of the mouse. So you see that the model-based image is actually much more quantitative than the optoacoustic image. The other good thing of model-based reconstruction, or sometimes it's called the iterative reconstruction, is that we can, in the inversion process, we can do more things. We can put, for example, constraints. Putting a non-negative constraints helps a lot to get rid of negative values that we're not interested in. We can also add regularization terms, which also help to get the image that we want. And this is just an example of how, for example, including this regularization and constraint terms can help getting better images. And also what you see here in this graph here in the right part of the slide is that the images are more quantitative. So what the line indicates basically the signal for a given insertion as a function of the concentration of the probe that you inject there, which should be linear, a signal with the concentration, but we see that if we use like a normal model base, it's not linear. But if we use this modification, we get much more quantitative results. Okay, so another thing to take into account when we reconstruct an image is effects related to the propagation of ultrasound. So I have here also two examples. These were the first papers that I was doing on this topic, or I mean my early work on this topic, where we what you see in the left-hand side is that a modification of the algorithm basically to take into account that the speed of sound inside the tissue is not exactly uniform, and it's not exactly the same as in water, which is the coupling medium between the tissue and the transducer. So you see that, for example, when we consider a uniform speed of sound, the speed of sound of water, the image that you see on the top is very much distorted. But then when we take into account the different speed of sound in the tissue, we have at least better defined structures like the spinal cord here and the vena cava. And another example of how acoustic scattering in this case affects the images, you can see it in the right-hand side of this slide. So this is an image of a zebrafish. A zebrafish is also a standard model in research. And you see that in the image in this one, in the left, you see that there are a lot of artifacts, and these are basically caused by reflections or scattering of the ultrasound waves, which are not taken into account in the standard reconstruction algorithms. So also, if we account for this, we get a much nicer image, I think, like the one that we have in the right here. And we can also distinguish, for example, the structures that are the fins of the fish that we cannot distinguish in the presence of the noise. Okay, so as I mentioned, so like ultrasound propagation is important to improve the optoacoustic images, but it's also important because we can combine optoacoustic and ultrasound. And the advantage is that obviously like normal ultrasound is a standard modality, particularly in the clinics. So people or doctors use it regularly, and they know how to interpret the image. Optoacoustics is a new imaging modality, so they don't know what. I mean, if you talk to a physician, then generally they don't know what to expect of the images. But if combining it with ultrasound is probably the way to go if we want to like implement this technique in the clinics at some point. So do you see this is like a particular specifically designed system that we developed to take into advantage to a specific array that takes into account the specific features of optoacoustics and ultrasound. So it's not a standard array, so it has a linear part for ultrasound, and we add these two concave parts to have this tomographic coverage that we need for optoacoustics. So for example, the images on the top, if you see that this image here is the image that we obtain in optoacoustics only with the linear part, which is clearly distorted, and it's improved with this array. While in ultrasound, sorry, it's the other way around. So the best image you get only with the linear part, because these elements have a very large pitch. Okay, so we can combine optoacoustics and ultrasound and get a lot of information. So this is another example of optoacoustics images. Here we have standard ultrasound, which is what we call here reflection ultrasound computed tomography. This is with a system that has like a full ring around the mouse. It's a different array that is enclosed in it's a circle around the mouse. And in this case, we can also measure different ultrasound properties like the speed of sound or the attenuation distribution in the tissue, which is also very useful information. And in the case of optoacoustic images, the images that you see here are actually very much processed. So apart from reconstructing, taking into account the different speed of sound and all these parameters, so we can enhance the contrast. For example, this is a different processing steps. This is like a histogram equalization step, followed by a Frangi filter. Frangi filter sometimes is a bit tricky because it makes new vessels appear, but the images look much nicer. And finally, segmentation of the image. So from this image, if we process it properly, we can get a much more enhanced image. Okay, so this was about 2D images. 2D imaging, we can also build systems that provide three-dimensional imaging, and this is an example of a system that I particularly like, which we call spiral volumetric optoacoustic tomography. The array in this case is spherical. It's a spherical array, also custom made. These arrays, none of these ultrasound arrays are standard. We have to make them, we have to specifically design. We don't make them ourselves, but we have to contact the company that specifically designs them for us. And in this case, so we have like an aperture in the array where light is delivered, and this generates, again, the ultrasound waves, and then we detect with all these elements here that you see independent signals. So in this particular one is 256 elements, but we also have arrays of 512 elements. And with these signals, we can make an image, not like this one that we see here in the left. For this, we need to scan the array all around the mouse. But if we focus in one particular area of the mouse, for example, the heart, we can see in real-time imaging, because only with one laser pulse we have sufficient number of signals to to build a three-dimensional image of a relatively small region. Again, we can use a standard back projection to do the reconstruction, but we can also develop a model-based approach in 3D, which is the same idea as the one in 2D, but the mathematics is a bit different. So, but it's just a matter of discretizing the equation to get here. Now this doesn't move. Okay, I cannot. This is blocked. Arm. Ah, thinking. Okay. Let me see if I can go back. Oh no.
I think that you need to share the screen.
No, but somehow PowerPoint just shut down.
Yeah, that also, but you need to share the screen.
Yes. It's sharing. How much time do I have?
Pero igual tienes que descompartir y volver a compartir.
Tienes como dos tips.
Sí.
Dos tips abiertos.
Oh no.
Es para aumentar las estadísticas de asistencia. Se abre varios.
Okay, I think we were here. So for whatever reason, this slide is problematic. It's thinking now. Oh no. Well, you can see it here, right? Maybe there's a problem in this slide, but I just wanted to show that in the case of 3D, we cannot with the 3D model, we can also account for the effects of the finite aperture of the transducer, the shape of the transducer, which is not a point, is a finite area, and model that as well. And this also helps improving the images. So this is what you see here is a comparison of like back projection and model-based reconstruction. If you zoom into details, you see that many vessels that are missed in the normal implementation of normal standard reconstruction, let's say, we can recover them with this model-based method. Okay, let me try this one. Maybe if I can share now. It's thinking. Okay, while it's thinking, one thing that I can tell you, it's that when we do the reconstruction, particularly when we go for a 3D reconstruction, this takes, well, a relatively large amount of time, right? It's computationally costly process. So it can be done in a CPU, but it's generally slow. And particularly if we want to have a preview during the experiments to have a real-time preview of what we're imaging, it is essential to do a GPU implementation. So this is what you see here is basically a bit of the description of the work that we published back in 2013 with the GPU implementation of a back projection. So basically, there are two steps. One is the filtering, and the other is the back projection per se. So in the filtering, we parallelize different signals, and in the back projection, we parallelize different pixels. And doing it like this, so we can get a very fast reconstruction around like 20 to 40 frames per second, and this is sufficient to make a preview. So you can see here, so this is a scan of a human, and if this video is working, okay, now it's playing, or it's oh no, PowerPoint was enabled. I have to say that I tested the presentation yesterday and everything was fine. So what do I do here? Yes, I shouldn't click yes. Okay, well, I can maybe just quick, it's not here. Okay, but you can believe it that it's something similar to this in the end. I mean, it's just, you can see in real time a 3D image like this, that basically this is plotted at different different views of the 3D image. So this is the XY view, the XZ view, and so this is depth. So the depth is approximately 1.5, maybe a bit more centimeters, and not more than that. Okay, so let me see if I can now share this. Okay. Okay, now it seems to be working, but still thinking. How much time do I have?
As much as you need.
Okay. This still doesn't work, right? Okay. Well, so the other thing, as I mentioned, is that there is an additional dimension that is the spectral dimension. So we can not only make an image, but the image is, we can make multiple images at multiple wavelengths. Basically, one image would give us the absorption at a specific wavelength, at the wavelength of the laser, but we can tune the wavelength of the laser. There are specific lasers for that that can that enable to change the wavelength in a given range. And then we can do like images, optoacoustic images at different wavelengths. And with this, we can not only basically determine where absorption takes place, but we can also know what kind of absorbers is there based on the spectral signature of different absorbers. For example, I mentioned hemoglobin. Hemoglobin has different absorption spectrum when it's oxygenated and deoxygenated, so we can distinguish between the two with optoacoustics, but also any dye or any fluorescent protein or any nanoparticle that has a given spectrum could also be resolved with optoacoustics. And for this, we need to do what's called spectral unmixing, which is an additional processing step to extract this information, which basically just consists in formulating the problem like this. So we have like the optoacoustic signal is given by the concentration of the different probes and the what's called the extinction coefficient, which is the absorption as a function of wavelength of different probes, and then multiplied by the fluence, which is the energy, basically the energy density of the laser. Yeah, so doing it like this, and also yeah, there are also different methods to like resolve the concentration of the different chromophores, which is what we're interested in. But there is a problem, is the fluence sometimes gives a lot of problems because it's not uniform. It also changes with wavelength, and it's very tricky to take into account this. So the first thing that we can do is to take it as a constant and then forget about it. But of course, we have errors with that, and we have to develop more sophisticated algorithms. So this is like the spectral part of optoacoustic, which is also very useful because we can distinguish any substance that has a defined absorption spectrum can be resolved with optoacoustics. Okay, so now we come to the last part of the presentation, which is basically the things that we're developing now, which is we're trying to push the limits of optoacoustics and basically in two different aspects. One is the temporal resolution. We want to image faster than what we do so far. I mean, the images that I showed, these real-time previews, these videos are acquired generally at 50 to 100 images per second, which is very fast for three-dimensional images. Not very easy to achieve this with any other modality, but we want to go faster than that. And on the other hand, we want to also enhance the spatial resolution. As I mentioned, optoacoustics provides good resolution because it's given by ultrasound rather than by light. With light alone, you cannot image with high resolution inside tissues, but if we take advantage of the ultrasound, of the low scattering of ultrasound, we can provide much higher resolution, but there is still a limit, which is acoustic diffraction, and then we're trying to overcome that limit. So I'll show you some examples. So one is what we call high frame rate of acoustic imaging, with this, which we could reach up to 1.6 kilohertz. And the trick to do this, I mean, our current bottleneck is the data throughput capacity of the system. So if we acquire many signals and many times per second, there is a moment in which there's too much data that we cannot handle. So what we do instead is to acquire only for every laser pulse only a given number of elements, for example, the red ones. Then the next laser pulse, we will acquire the yellow ones, and the green ones, and then the blue ones. So we split the elements that we acquire into different pulses, and then we can take into account that in the reconstruction. So we not only reconstruct an image, we can reconstruct the whole frame simultaneously, and we add what I call here a total variation regularization, both in the space and in the time domain, because we have this information that is spatial and also, I mean, with time, it's a sequence of images in the end. And I'm going to show you an example, which is, I think it's very nice. This is a freely swimming zebrafish. It's a zebrafish larva, very small. So the image is not I mean, it's only a few pixels, but you see how it's swimming. And I can play it again. I mean, this is super fast. Actually, this is not real time. You see that the whole video is right now is 200 milliseconds, and I think the whole video is less than 1 second. So if you see this in real time, basically you see just moving too fast the tail when the fish is swimming. So the video is less than half a second, the total video. But we can resolve it with optoacoustics. Deep learning, of course. So you're, I think you do some deep learning here. Or some artificial intelligence, and it's yeah, we also did something about that. As I mentioned, when we go for a high frame rate imaging, one thing that it's important is to reduce the amount of data, which can be done, for example, reducing the number of channels that we acquire with every laser pulse, and then we have to do something to make the image better when we don't have enough information. So we also did that with deep learning. So this is an example that you see here. These are the images that we acquired with the 2D system. This is a cross section of a mouse. This is a nice image because we have 512 elements, 512 sensors. But if we reduce the sensors, so I think this is 128, and this here is only 32. So you see that the artifacts are dominant in the image, particularly in 32 with only 32 channels. We barely can see anything when we do a standard reconstruction. But what we did here was to train a unit, which is a standard, I would say, now it's kind of a standard neural network, to recover this information. And for this, we use the good images provided with the system with 512 elements, and the bad images provided with only 32 or 128 elements. And you see that this is the result. So basically, it's very, I would say, amazing what the network does. The other thing is whether it's true or not, but at least from something that looks like nothing, it makes an image. And here, from something that looks with a lot of artifacts, it makes a clear image. Now, as I mentioned, this is very nice. Now, relying on deep learning, I have my concerns whether this is true or not, what we're seeing there. Because even though here we have very nice data to train, because we're training actually with real data, other groups have done the same, but with simulations. But even having good data for training and a lot of data, of course, if you find something different, what would the network do? Nobody knows what it does, right? Okay, so another different approaches that we developed also recently to do imaging with only one element. So this was only with a few elements, as I mentioned, 128 of 32 elements, but we can even try to push it further and try to get image like an optoacoustic image with only one element, only one signal. And a trick that we did in this paper here was to use, I mean, this is it's an array, but actually we're acquiring all signals simultaneously, and we only take one. So we put here a random distribution of scatterers, and this basically kind of codifies the position of the source. And with this, we can make a model matrix that in the end we can use to make a reconstruction. Of course, it's very tricky to do this, and we only image a very simple sample here, like a zebrafish larva that you see here. But we kind of recover the image with only one signal. So this image that you see here, this is the image that we obtained with the full ring, I mean, 512 elements. And this here in the left is the image that we obtain only with one element. I mean, it's a very, very simple image. As I mentioned, this is just a few pixels, but still the fact that we're able to reconstruct with only one element, I think it was very nice. And another example of only imaging with one element is shown here. This is based on PCA, which is also considered or can be considered a machine learning approach. And so it's also based on training. And this image that you see here, or it's actually a video, if it works, of course, this is the beating heart of the mouse. And it's a very periodic motion, and this is the advantage of this. So basically what we did here was to train with PCA, I mean, used as training data the images acquired at the low frame rate, but a lot of images. And since the images are all look the same, so in the end, when we do only with one signal but at higher, or with less number of signals but at a higher rate, we can still reconstruct the heart, which of course it's impossible if we do it with normal back projection or normal reconstruction. So here in the right part, you see this is only one signal, and we even see something with one signal. I mean, of course not perfect, but we kind of still recover the heart. But again, this is also very tricky because it's a very periodic motion, and it's also not a general case, but still it's very promising that we can do imaging only with one signal. Okay, finally, we come back to the part which I'm mainly focusing on right now, which is super resolution imaging. So as I mentioned, with optoacoustics, we can provide much higher resolution than with optics, but we're still limited by acoustic diffraction. And we can do some tricks to overcome that. So one approach is what we call localization optoacoustic tomography, which is based on localization imaging. I don't know if you heard about this, but this also a technique that was awarded with a Nobel Prize in the case of fluorescence microscopy. And the idea is that when we have isolated sources, we can localize it. We can localize them, basically meaning determine their position. And localization of an isolated source can be done with a much higher accuracy than distinguishing two sources, which is what's called the resolution. So basically, if we have isolated sources and then we move these sources, what we can build is an image that is basically a set of points in the end. But as I mentioned, the localization of these points is very, very accurately. So when we build an image like this, we can actually get much higher resolution than the one that we have originally that is dictated by diffraction. So you see here, this is a comparison. This is just a phantom where we injected some particles, and the image in the left here is the image that we get with super resolution, and the one in the right is what we get with standard optoacoustics. You also see in this case that there are some missing information, which, as I mentioned in the presentation at some point, this is because of limited view. And with this technique, we can also overcome this, which is, I think, it's also a very nice advantage. So of course, this is a phantom, and very recently, now we finally managed to do this in vivo. Actually, it took us a lot of time, and I'm going to show you the difficult part. Basically, to do it in vivo is to develop some kind of particles that you can detect in the presence of blood, because blood absorbs a lot, and the particles have to be super absorbing to absorb more than blood and appear in the image. So, but I'm going to show you here. This is the brain of the mouse, basically what you see here. And now inject these particles, and you'll see that you will see them in the image, flowing in the image, basically. So now what we can do is to localize these particles, or to track these particles, and build an image based on that. And this is what I'm going to show you here in the right. So you see that this is the super resolution image where you see much more details of in this case of the brain vasculature. And finally, this is the last slide. I know that well, we started late, right? Yeah, so we can also do super resolution imaging with some tricks in the reconstruction, particularly when we do some regularization based on the L1 norm. So we also have shown that it's possible to go beyond diffraction like this. So you can see it here. This is an image. Basically, this is just two absorbers. Well, the one in the left here is just one single absorber, where you see that. I mean, the full width half maximum is lower when we use this approach than when we use standard approaches like the SAFT or the normal reconstruction based on L2 norm. But if we have two absorbers, we cannot distinguish them with standard approaches. But if we use the super resolution approach, we can see like the two dots here that correspond actually to two absorbers. We also tried this approach in vivo. The advantage of this, of course, is that here we don't need to inject anything. This is just based on endogenous contrast, which makes it much more flexible. But it's not so, I mean, the enhancement of resolution is not as good as with localization, but still it's pretty good, I would say. And so we can improve the image also with this approach. So that was it. So as concluding remarks, just a couple of take-home messages. I hope that I have convinced you that, or at least the message that I tried to convey was that there are many optoacoustic imaging systems. We have to design both the hardware and the software components, and both have to be taken into account to get good images. So this is actually what you saw here. It's a lot of work, many years. And there are the last part of the presentation where I show you the new methods that we're working in now, so we can show that we can break through current limits of spatial and temporal resolution with new approaches that we're developing. So finally, I have to acknowledge the group. So you see here, this is the boss. This is Professor Daniel Rasansky. I'm here. So this is all the group members. And I thank you for your attention. And I will be very happy to discuss with you and to answer any questions. Actually, that's the part that I enjoy the most.
Thank you very much.
I mute.
Maybe we can try and if you can mute your computer. Again.
Okay, no. Está muteado el tuyo. Did you mute your...
It's muted.
It's muted.
I can
No se puede hacer nada.
now,
I can...
so now,
Yeah, hello? Hi. I think you can hear me, right? Yes.
Yes.
And,
Yes.
okay. Yes. Okay, so thank you very much, José Luis, for your presentation. I would, if you allow me, I would like to do now something I didn't do at the beginning because we just rushed into the presentation because of the lack of time on the delay. But I forgot to mention, to introduce José Luis properly, that he, I mean, his role right now, he's a senior scientist and he's also group leader at the University of Zurich, at the ETH in Zurich. So he's a very renowned scientist in this field. And I have to say I was very much impressed by the results he was showing. Particularly, maybe you mentioned, and I lost it, what is the spatial resolution that you achieve with this ultrasound technique?
yes, I mean, generally the resolution, I mean, as a rule of thumb, I mean, as I mentioned, there are, can you hear me or?
Yeah. No, tienes que volverte. You need to, yeah. I need to.
Can you hear me now? Now.
Sí.
Okay. Yes, the question, I'm going to repeat it just in case, is what's the resolution that we can achieve? Yeah, so as I mentioned, there are many different optoacoustic imaging systems. So basically, the resolution depends on the depth. If we only image superficially, as I mentioned, we can focus light. This is very high resolution. We can even go to micron level. If we go deep, the rule of thumb is generally 1 divided by 200 of the imaging depth. That means that if we go 1 millimeter, if we go, let's say 1 centimeter, that would be around 50 micron resolution. But depends on the system that you use. I mean, the typical resolution that we get is, 100, something like this. With a standard optoacoustic image, 100, 150 maybe. If we go to this super resolution method that I described, so with this, we can push the resolution. We can get even 20 microns or even better than that. Yeah. Okay.
I wonder if there are other questions from the people which is participating. You can also use the chat if you prefer. I guess.
I have one question, one information and one question. The information is that Xosé is from Galicia.
Exactly.
Yes, he speaks very well English, but he speaks also very good Gallego. And the question is the following: are there any commercial devices? And yes. If that is the case, are these commercial devices used in clinical applications?
Yeah, that's a very good question. So I didn't put any slide about the clinical translation actually, but I mean, there are commercial.
You need to connect your.
Yeah, sorry about that.
No, that's fine. Okay, yes. So the question was whether there are some commercial systems, whether the technology is commercialized, basically. So it is. There are different companies that commercialize. If you just Google optoacoustic or photoacoustic, you will find also some companies. One of them was actually built in Munich when we were at the institute there. I mean, I'm not involved in in the company. I have no interest with them, but it originated as a spin-off of the institute in Munich. They're actually very successful, and now they're also trying to commercialize the system for clinical use. So for clinical use is a bit more tricky because they need the CE certificate, but they got it recently, and now you can even buy a system that is clinically approved to be used in a hospital. And regarding the clinical use of optoacoustics, I mean, since I would say 2015 or something like that, there were already some clinical trials of different things. The image, skin conditions, that's one thing. The image also breast cancer. What else? Metastasis in the lymph nodes and other applications. Also Crohn's disease, which is a disease in the intestine. So it's actually pretty deep, and it's not easy to reach that deep, but they manage also to get something with that. But and these are clinical trials. I mean, our initial clinical trials, but these are with real patients. So we didn't do it ourselves, but there were different groups in the world that did it. Yes. So I think in the future, probably it will be more things in the clinic will appear. I'm mute again.
Personally, I think I see lots of connections with different things which are being done at CiTIUS. So as you mentioned, deep learning, but also more specifically this GPU programming in order to speed up computation, because you mentioned you were getting real-time processing with 20, 40, up to 40, I think, frames per second, which is quite impressive for this kind of volume of data. But also from a more, let's say, imaging point of view, I'm just wondering, you show a very nice setup, custom made, I guess, with different ultrasound devices with different wavelengths, so green, yellow, blue, and so on. And if I understood correctly, you were acquiring the images by doing different shots at different, so for the different wavelengths, so at different frequencies, let's say. Have you thought about going one step further and trying to do a one-shot multi-frequency acquisition? That would be more complicated from the signal processing point of view, but at least in the optical range, this is complicated but doable.
Well, I don't know if that is possible, because in the end, I mean, every image, I mean, every excitation, let's say, is a very short pulse. It's like a nanosecond duration pulse, okay? Now, the question is, when do you, I mean, you can, of course, let's say, potentially mix different wavelengths into the same pulse, although it's not possible with the laser that we have, but let's say you have two lasers and you mix them or something like this. Then the problem is how to, I mean, distinguish the two of them, because you're putting both at the same time. Now, the other question is, if you want to separate the pulses, how long should you separate them together? So you can, for example, like put the two pulses and acquire this, like in one acquisition, get already the information from the two pulses. Actually, we did something like this, so the pulses were separated by, I think, around 17 microseconds, and we needed that because sometimes when we move too fast, even the distance between pulses is enough to get different images, because I mean, you have motion between the frames, and then you cannot do anything basically. But if we put the pulses very close, we can also like have everything together and then mix it.
Mm-hmm.
Yes.
Okay,
I can show you that.
Yeah, that is very interesting indeed. So is there any, I think, no, I don't see any raised hands. Okay. I think, okay.
Very different from...
Not really. I think we have to get rid of these Teams tools, I think. Okay, if there are no further questions, please let me thank the speaker again for his presentation. And well, thank you everybody for attending the presentation. Thank you.
Al menos que te lleves los aplausos aquí. Los despachos es difícil oírlos.